This is the second webinar in a series of webinars that have been run by Pharmacometrics Africa across the whole of the year 2019, going forward into 2020. And Pharmacometrics Africa, I'll give you a brief about it, is a not-for-profit organization whose sole goal and vision is to increase the number of trained pharmacometric scientists working on locally relevant data on the African landscape through a series of training activities. So a few points to note for this webinar. If you have any questions during the course of the presentation, please write it down in the chat and we will collect these questions and let our speaker answer them, or you'll be answered in the chat. And so yes, please feel free to use the chat function. At this juncture, I would like to introduce our speaker, and it's my pleasure to do so. Our speaker today is Dr. Joel Owen. Joel is the VP Strategic Modeling and Simulation at Cognigen. He's an adjunct professor at the College of Pharmacy, Union University, and he has a wealth of experience with more than 25 years working in the modeling landscape in both pharma and in academia. His research focuses on application of quantitative methods for the development and use of therapeutic compounds in different disease areas. I believe that we are going to really benefit from the wealth of experience that Dr. Joel presents. And right now, I would like to hand over to him to take us through his presentation.
Thank you, Ida. I appreciate the opportunity to be here and to share today. I'm going to share my screen and get started here. So the topic of conversation today really is about data integrity and the importance of data integrity and having your work be credible and be reliable, so that when you publish or when that information is used for regulatory types of decisions, that we know that the work actually represents what the data are telling us. And I appreciate the opportunity to be here. The agenda, I just want to give a little bit of introduction to some data integrity kinds of topics, and we'll talk just briefly about regulatory agencies. I think our audience primarily is academia. That's how we focus this, but sometimes our work, even in academia, winds up in regulatory settings and may have an impact that's larger than what was originally intended. And so we want to pay attention to our data management as we go through. I've got a case study that's I think is a very interesting and relevant one that it comes from a practice in Uganda, and I want to convey that in a little while. The idea of canonical documents we'll talk about, and then some data management specifics, and then the idea of a data repository and how that can be very helpful. So data integrity implies a number of key characteristics about our data. When we think about data integrity and data management, it really is a discipline in itself. The original data are an important part of that. So some of the characteristics that we think of are the originality of the data, the accuracy of the data, are the data attributable, that we know where it arose from and that it's sort of its life cycle. Are the data verifiable, and are they complete? The WHO uses an expression, ALCOA plus, which takes the traditional attributable, legible, contemporaneous, original, and accurate characteristics and adds to them completeness, consistency, and enduring, and that they are available. So these are principles or characteristics that we need to make sure that we have systems in place to manage as we are working with data sets. So a lot of data management and data integrity issues certainly arise in their sort of to their highest importance when we think about the development of new drugs. The highest expectations of our data integrity are when information is going to be submitted to one of the stringent regulatory authorities, such as the FDA or EMA and others. In academic settings, though, we may have lower sort of documentation requirements. We don't often have someone coming in and performing a a review of our data management processes, but we still want to practice good data management, because sometimes the things that we do can wind up with a higher purpose that would lead to greater scrutiny of the data. So we want to, in the context of the pharmaceutical industry, a new drug has to be developed and tested in accordance with stringent regulatory regulations, and approval is based upon the data collected and reported, and audits and that sort of thing can happen. An example of those regulations in the United States is the 21 CFR Part 11 compliance regulations from the FDA, and they require a great deal of paperwork, standard operating procedures, a lot of process goes on to ensure version control of programs and data, that there is appropriate archiving of electronic data, that it is adequately backed up and stored. And these regulations particularly speak to electronic signatures and the processes that are required for that. They also speak to the validation requirements of computer systems and software. The ICH also has its own expectations. So the International Council for Harmonization of Technical Requirements for Pharmaceuticals and Human Use is the ICH effort. So we're trying to, in this process, align regulatory standards, but they have their own series of technical guidelines and insist on high quality data management. The World Health Organization likewise serves as a stringent regulatory authority that regulates the approval of drug products in quite a number of countries, and they have their own regulations. There is an effort, and Pharmacometrics Africa is playing a role in this, to shift the center of gravity for African science to Africa. And I think that having high quality and reliable data and data management will be an important part of, to have a credibility of research results, that the data management is going to be an important part of growing the respective research and publications from African sources. If we think about data management, there's a number of stakeholders that are involved. And each group may have their different perspective on what is important about data management. In an academic setting, we think about advisors who are mentoring students, and those students who are doing work, and if that work is sponsored by a drug company, the sponsors have their own concerns. Clinical investigators and regulatory agencies each have their own perspective on why data management is important. And even when you go to publish the work, the data quality is going to be important. A few questions we might ask ourselves as a student, and why I care about the quality of my data. Can I replicate an analysis and know my work is correct, would be a question perhaps. So if we are doing work and we've got it on a laptop and we did a bunch of manipulations in Excel and we save that Excel spreadsheet, and can we know in the end that we didn't make a mistake along the way in using an Excel spreadsheet, that sort of thing. An important question to an advisor perhaps, and I've seen this myself, is we have students working for us and they are building a data set and they've done some work, and perhaps they move on to another site. They graduate and move on. And do I know where the data are as my student has gone on and left with their own information? Sponsors will certainly be, if work is going to be used in any sort of regulatory setting, the sponsor company is going to want to know that, and the funders, that the data were developed appropriately, they were managed appropriately, and they have all those proper characteristics that we talk about.
Joel, I'm going to launch the poll right now. Okay. So please, right now, please vote. You're going to shortly receive the questions on your screens.
So the first question is, have you ever lost data that resulted in a setback or anxiety in your project? This might be a hard drive crashed. you stored it on a cloud drive, and it was on a hard, you were looking for it on your hard drive, other such things. So the second is, have you ever had a student or a collaborator working on a project leave your lab and take their work and their knowledge of the data with them? Can you reproduce what someone else did who was one of your collaborators? Has your work ever been used in a way that it wasn't originally intended, would be the third question. And if you can answer those, well, how about the folks in the room? Has anybody ever lost data or had anxiety for a little while that you thought you had lost data, or couldn't count on the quality of a particular file? Yeah, I see a few hands, so I've been there myself. For the second question about having to keep up with data from a collaborator, does that ever pose any issues? Yeah. So that's certainly been my case as well. I see the advisors in the room raising their hands. Has anyone ever had work used in a way, so you've done a project, you've found certain results, and then you see them sort of take on a life of their own, and they carry a meaning or some impact that maybe is beyond what was originally intended? Anybody had that case? Yeah, great. So. And I see some yeses and nos from the online poll, and that's great. Okay, so my case study is one example where a project did sort of take on a life bigger than, or an impact bigger than perhaps was originally intended. There was a study of efavirenz pharmacokinetics, pharmacogenetic implications on the kinetics of efavirenz that was done in Uganda. Jackson Makanzo was the advisor and the first author on a couple of the papers. This was funded by a couple of organizations, but originally didn't intend to have any, at least I don't know that it was expected that it would have regulatory impact. This work was conducted in Uganda through Makerere University. And there's a couple of the publications noted here. After this work was done and the papers were published, the results were presented at the Uganda National Pharmacovigilance Workshop, and a couple of organizations, including the Uganda National Drug Authority, expressed interest in the findings. So there were individuals at the workshop that saw the presentation and found it to be an interesting information. And so they invited Dr. Makanzo to come and present at a couple of meetings at the National Drug Authority and the Ministry of Health Uganda offices. And they began to talk about, based on those results, changes in the HIV treatment guidelines for efavirenz. And the issue was a lower dose is perhaps needed often because of CNS side effects, and the guidelines didn't allow for that. Kind of simultaneously, the World Health Organization was already investigating this themselves, and in 2016, this was kind of running, these were running concurrently, the development of these papers and the guidelines, but the World Health Organization did recommend a reduced dose of efavirenz to improve tolerability. And so they've written a nice guideline. There's a list of references there. Though Dr. Makanzo's work isn't listed in that reference, it was, and that was really an issue of timing. The work was referenced in a presentation of the new guidelines by the World Health Organization at a particular meeting in 2016. And the prior research and meetings that Dr. Makanzo had really fast-tracked Uganda's policy decisions, and they had already moved towards having a 400 milligram dosage form available. That was the lower dose. And they had already begun to set up a plan to collect data on special populations. So this is an example, I believe, an important example of some regulatory decisions, some guideline changes being made based on a literature result from an academic setting. So one thing we should learn is perhaps to always assume your work is important and will have impact. Even if it's preclinical kinds of work, we're generating data that might feed into a PBPK model or something that would could eventually work its way into a model that affects human dosing. So regardless of where you are, your work could be important. So you always remember your work may make a significant contribution to patient health. And because of that, we need to pay attention to data integrity and the quality of our data. The idea of canonical documents is an important one, and by the canonical documents, we just mean a canon of a few particular documents that help us maintain quality in our work. So they are core documents. They're almost always present when we're doing things in an industrial setting and we're doing work for drug approval, but I think it's wise to develop some of these practices even in an academic setting to create a specific statement of our processes. And we do that to aid in quality and consistency of the documentation. It does require careful analysis of what we're doing, what our delivery products are going to be, and we can define some common elements of what ought to be in some of these documents. They can also be a part of developing a strategy for improving the quality and reliability of our work. The canonical documents that we see as important really are a data analysis plan. So we want to in a written document define what are the study details, what kind of, if we're analyzing a PK study or a series of PK studies, where do those data come from? What are the endpoints we're going to assess? What types of PK or PD measures? We want to think about how we're going to use the data and what kind of modeling is going to be applied, and go ahead and specify, sort of pre-specify our analysis, and then define what our deliverables are going to be. Most often in an academic setting, that's going to be the development of a manuscript. A data management plan is something that we overlook a lot, and it probably is rarely done in an academic setting, but to know what kind of data you're going to collect and have a plan for how it's going to be organized, where it's going to be stored, where it's going to be backed up, how items are going to be defined in the data set. Those sorts of things can be very important to maintaining the quality of a data set. And then we want to think about technical reports and manuscripts, how we're going to structure the report. Now, often this is going to come from guidelines from the publisher. What are the sections going to be and all of that, but we want to think about how we're going to state and define what was done. And as we go through that process, we want to be thinking about the data that we're working with. What do we have to define the analyses that are going to be performed and reported? When we think about regulatory accountability, we need to have documents that describe the data themselves, what items are in a data set, what are their units and those sorts of things, where are they stored, and there need to be appropriate labels on the variables. So I just want to give a couple of examples here of maybe some poor practices or some good ideas on how to do things better, perhaps, and things that we have learned along the way in our work at Cognigen and my academic work in the past. So this is an example of a data set that was shared in, this is a modified version, it's hypothetical data, but data come like this often from bioanalytical labs where the results are presented in Excel, and it's not an absolute rule that you shouldn't use Excel or spreadsheets to record and work with data, but it's not the most efficient way, and it's not the best way to document the quality of data. We can make very nice, pretty tables and things with our Excel kinds of spreadsheets because you can do such nice formatting. But it makes it very difficult to use. So if we think about how data are going to be used after they're produced and recorded, we need to think about their format and in the way they're going to be used. So if we needed to analyze these data if this is how they were reported to us in any programming environment, these are going to be data that are very hard to use. So one example here is they've used highlighting to indicate some places where data were interpolated, and one might want to keep up with that as a record. The overall layout here, we have times down the left column, and then these are just different periods of PK collections. And so if we needed to read this into win non-len or non-mem or something like that, the structure is very hard to manipulate and get into a useful data set. So having comments and colors and things are nice in Excel, but they're really not able to be used very well in a programming environment. This would be a better way to present the data. We would call that maybe reusable data, because you can present the data here. It's maybe not as easy to read for a person to look at, but we can now read it with a computer program. We want to have like items in their columns, and we can add to the data set additional imputation flags and such things. So we can add additional variable items to the original data. As long as we document what is there, but you keep the original data and then have perhaps an imputation flag, a new variable item that says this item was imputed a value because the value was missing. It's also a very good practice to have data labels, so that we store and know what the formats of the data are, and then have labels to define their units and things. And this is an example of a SAS proc output, and it tells you the variable names that are in the data set. To have this kind of structure makes it easy to communicate and understand what's in the data set. Another really important practice, even if you're working in Excel, but in any environment, would be to have a data set that is sort of self-documenting. So we have a decode. So we often need numerical items, particularly if we're working with non-mem and certain analysis software. The variables that we're going to actually analyze need to be numeric and not character. But if analyte is in the data set as a 1 and a 2, then somehow we need to know, even 2 or 3 years when we come back to the data set, we need to know what 1 and 2 meant. So to include an analyte character, or add a C to the end of the name, and then actually define what those numbers mean in the data set, then it's a self-documented data set. You don't have to have something extra stored along with it. Is a good practice. So up to this point, we've been talking about really acquiring data. When we think about analysis data sets, a good place to start is reviewing the analysis plan and thinking about the data that have been collected and how they are going to be used. So we want to review that plan. We want to review the source data before we build the analysis data set and know that we have everything that's needed. We want to understand the key fields. So often we're getting data from multiple sources. We get case report form data from a clinic. We get bioanalytical data from a laboratory. Those have to be merged. So we need to make sure we understand the key fields or the merge variables, and that they are each data set that's being merged has the appropriate items into it. And we always want to keep the source data as unprocessed. And it's a good practice if your operating system allows it to receive the data or develop the data and then make it uneditable through at the operating system level, so that it's not accidentally overwritten. That's easy in Unix. I'm not sure in Windows, but keeping the source data so that there's a documented source to always go back to is an important thing for accountability throughout the process. And it's a very good idea to use a programming language such as R or SAS to actually build the data set. So if you can do your merges and your reformatting of data, whatever needs to happen in a programming environment, then if you have locked source data and you've kept the programs, you can always repeat and know every step that happened along the way in building your data set. So it's really a better approach for quality data management. Is if you're working in a programming environment to do your data management. You can use Excel. You can do everything the same in Excel. It's just more manual, and it's easy to make a mistake, and there's little traceability on the steps that are involved. Another example, so if we're moving over to an analysis data set, we've seen data sets like this occasionally where the actual measures for the analysis are the means of multiple measures. So these values, those numbers are pretty small, but the values, the assay was recorded in triplicate, and then it's the mean of those values that's going to become the analysis variable. Well, in this data set, they were provided in cells were merged in Excel. So to actually read across the data becomes a challenge if we're going to kind of do these merges. So if we can not merge cells in a spreadsheet kind of environment, if we can do things programmatically, we wind up with much more useful data in the end. So a data set build should also always be appropriately documented. Every change you make to the data set, it's a good idea to either have the program that made those changes saved and stored, have units documented where you're changing units, make your process be apparent and be documented so that others can come back and have confidence in the work that was done as they review it. It's a good idea, again, as we are working with an analysis data set, that before we even begin any modeling or anything, that we do exploratory data analysis. We can often find errors in our data set if we plot the data and begin to explore minimums and maximums and such things as we go through the data set. Archiving and preservation of data is an important thing. We want to make sure that we think about how data might be used in the future. So if data are going to be stored for an extended period of time where software versions might change, we want to save the data, the analysis data, in formats that would be readable by future programs. So if CSVs are going to be readable forever, they're ASCII files that are sort of generic and anything's going to be able to read them. An XLXSX, whatever the format is, may not be readable by future versions of Excel, probably will be, but you're not guaranteed. Similarly, in SAS, use SAS transport files that are much more useful across different versions of SAS and that sort of thing. Also make sure your data are uniquely identifiable. Even in your file names, use file name structure so that you've got you can clearly identify what the data are and where they come from, not just mydata.csv. Have a meaningful name to it so you'll know what they are. And then you want to back up data because the unexpected does happen sometimes. So you want to have, if you can, multiple places of storage of at least the source data. So keeping it with good quality or redundancy perhaps in the backup, and off-site backup is a good thing. It's good to store relevant metadata, and by metadata I mean the date time stamps on files and such things, so you can account for the history of the work. And then also to store all of the documents that would have been related to the source of the data and how the data were analyzed, so protocols, analysis plans, the technical reports and manuscripts. So have a plan for storing the information. Adopting proper security protocols is important. So how do you keep your data secure? In the United States, we have a set of regulations called HIPAA, and it's just the ethical guidelines on how we blind individually or individual information so that individual subjects are not, or patients are not identifiable in our data sets. We're appropriately dealing with the privacy of individuals. So we need to make sure that in our data don't identify individual patients or subjects so that someone could go back and find that person. We need to keep data de-identified at the point that we can, and then keep it secure so that we're never jeopardizing anyone's privacy is one thing to think about. But making sure that others can't inappropriately gain access to data is important. So in thinking about structuring data sets, I've got about 3 slides here that look at just a way that we lay out and organize data. So as we go through studies and we think about running a clinical trial, we collect the data, we're going to go through analysis processes, we generate a lot of files along the way. And being able to, a part of good data management is keeping those in a structured set of directories or some way of keeping up with what the files are, and to develop your own sort of system for organizing those can be very helpful. So we have, there's a few slides here that are just some examples. We would store files at one level, sort of at a drug level. So what is the information we know about the drug itself? There may be documents that are sort of project or excuse me, information related to the drug. We might have literature examples and communication with regulatory authorities. These things are happening at a drug level. Another would be, as we think about particular clinical trials, we would have a way to organize and store the documents from a clinical trial. So a folder for case report forms and the clinical study report, protocols and such things. And then as we receive data, because data are often released in multiple iterations, we might get bioanalytical data in batches and that sort of thing. It's really important to organize your data so that you can know which version you're working with. We deal with that in part by having a separate folder for each time there's a data release, and we date the folder here in a way that we can see if we're going back into analyze, we know where the data came from. And then when we go to a modeling project, we set up projects in terms of stages, and we have data assembly and data set, the finished data sets and programs where we're running our analysis and things. And so having some sort of structured data set file storage system can be an important part of good data management, just because you know what the locations, the time sequence, and all of that is. The idea of a data repository is also a very good one, particularly when you're collaborating between groups. And Cognigen has set up a data repository system that it's really web-based for any academic sites that are interested in having access to something like this. If you'll let me know, we can, through some support from the Gates Foundation, we can allow some academic sites to have access to this particular package, but this is really just an example of how you might work in a kind of a data repository. But a data repository would be a way to share information among collaborators, but also to control who has access to which parts of the data. It has predefined directory structures, as I had mentioned, and there's control over the data, the metadata, the time date stamps. It really helps one be regulatory compliant in your routine practices. There will typically be an administrator who grants permissions at certain levels to different data. We can structure that, the data repository, really at again as a drug level, at the study and the project level, and these are really relational in their data structure and storage. And the data can be shared between drug products if we're moving from a single entity to dosing combination products. And it stores information appropriately. It provides a very good way for collaboration to occur, because we can share in real time. We can share data as they're available, and it can be controlled to certain team members having access. When data are uploaded into the data repository, there's a complete versioning and storage of the different versions of the data that are uploaded. In our data repository, we have something called Explore Live, so that you can click on a certain icon here, and it will put you in a system that will allow you to do exploratory data analysis and graphing and data manipulation to do some of that exploratory data analysis that's a part of good quality data management. So I think data integrity is key to the reliability of your study results, and having confidence in your findings and as they support decision making. I think that the growth of financial support for research will grow in part as results are have integrity and grantors have confidence in your ability to do work and to do it with good integrity. And it does take work and planning and systems and discipline and skill. Data management really is a skill and a discipline sort of in itself. And implementing processes that assure data integrity may improve your sleep if you're not worrying about all those issues at night. My slides, I would like to acknowledge that Ted Grisella, our president at Cognigen, and Rebecca Humphrey and Kelly Maxwell also helped in preparing these slides and thinking about some of these issues. So that's
Thank you, Joe, for a very stimulating and detailed discussion over there. I would like to open up the session now to questions. So right now you're able to unmute yourself, and if you have a question, please move to the lower left hand side of your laptops or computers and unmute yourself and ask a question. In addition, you can also use the chat function. Please feel free to ask a question using the chat function. We'll collect those questions and then give a chance to Joel to answer them. So the floor is now open. You can ask questions.
What would be your recommendation for students to back up the data? Because the reason why I don't do really often backup, because it takes a lot of time. And should I zip all my documents and put it somewhere else or something like that? But then it's super time consuming. So do you have any tips or recommendation to help students doing backup of data to be sure?
That's a great and practical question. How do we back up data in an academic setting that is not burdensome and also is reliable? So there would be a number of ways to do it. Again, you need to maintain, however you're backing it up and where you're storing it, you would need to make sure that it's kept secure and confidential or whatever. But if you have a cloud storage backup in some way, that can be most of the cloud storage systems, Dropbox and others are, they've got redundancy in their backups and all. On a local system, you know, making a zip file and putting that on a USB drive and then making sure you know where the USB drive is, that's a way to make an easy sort of off-site backup. If you have multiple, I mean, you'll have to work with the system you have at hand and what tools you have at hand. And I don't know that there's always an easy solution and a non-time consuming one. It's just one where discipline is, to remember to do it is kind of what's important. I don't have an easy solution for that. The things you've mentioned are good, but an advantage, if you've got a secure cloud system where you could back up on a Dropbox or something like that. I was asked if a Dropbox was safe. So you'll definitely want to look into the security of the system you're using. I do know, I've read the documents. The FDA uses OneDrive from Microsoft, and they have published in PDF files. They're available online. Their security and validation system that they went through. So I'm certain because of what's publicly available information, I'm sure that the FDA is using OneDrive, and they see it as a secure enough system that they're using that. You would just want to be careful where you store that. But in each of those sort of major commercial cloud systems, they have redundancy in their backups, and so it's not likely that you're going to lose data because a hard drive fails and that sort of thing. But it is a critical issue if your data is only stored in one spot and it's on your laptop and somebody steals your laptop or your hard drive crashes.
Now, this is not so much a question, but it's a comment on that. So academia and presumably public authorities have come to the party quite late. In fact, traditionally, we push back on all the sort of things that Joel has been talking about. Basically, we thought that was something which looked very ugly. However, any of you who have applied for any grants and so recently will now know that as part of that process, you have to actually put in a data management plan which will include all the aspects that Joel talked about. And data security is very important, particularly if you're dealing with patient data. And there will be protocols from local health authorities based with that. So it's taken time, but it is actually happening now in an academic setting. And so it's not any longer just a desirable. It is actually now becoming mandatory.
Thank you.
So while that question's coming through, I have one here in the room.
Okay.
We heard you talk about Excel and the problems with Excel. Could you give any anecdotes or examples of what could go wrong if you use Excel as your primary method of both storing and manipulating the data?
Sure. One of the main issues is just the lack of accountability. If something changes in a data set, not to say that I work when I'm sleepy, but I've worked in an Excel spreadsheet before, and you strike a wrong key and the cursor isn't where you thought it was, and you change a data point and there's no record of that. You don't know that something's actually changed, or there's a possibility in cutting and pasting that you make an error and put something in the wrong spot. So it's very hard to trace the history and the activity of the data. I have found just last week that they actually have added track changes to Excel like they've had in Word for a long time. So there, if you remember to turn it on, there is a track changes so you can see edits as you go along. But that's only if you turn it on. And then that's not necessarily reliable as if you had locked data. And then the other thing is it's just easy to do poor formatting, and then it's difficult to read those data in with a program where you would have a better history of how the data were changed as you go from recorded and acquired data into an analysis data set.
So Joe, we have a question from the audience. Adenei from Nigeria is asking, are there any open source data repositories that you know of, and which ones do you recommend? That's question one. William is asking, in your experience, have you ever handled missing data, and how have you done that?
Okay. The issue of open source data repositories, there are a couple that are available. There's one called OSF, the Open Science Foundation, I believe. It's not structured. It's not like the Cognigen data repository that I showed the examples of, but they do have a way in a cloud-based system to store and account for histories of data and that sort of thing. As I was doing a little bit of research in preparing for this talk, I did come across there's an African Science Foundation data story. It's like the Open Science Foundation, but it was focused on African universities and that sort of thing. So I don't know the details of that, but that could be Googled pretty easily. In terms of imputing data, so if we have missing data, do we try to account for that in some way so we don't lose subjects from an analysis? And that really is always a sort of a situation dependent kind of decision. But whenever we're going to impute data, that's one of the things we ought to think about when we're writing our analysis plan, and pre-state the rules of how we're going to impute data. So if someone has a missing body weight, for instance, and we don't want to lose them from the data set, one might impute the gender-specific median of body weight or something like that. You have to consider carefully what imputations are going to be made and how they might impact the analysis in the end before you do that. But imputations are often made not in the dependent variables, but in the independent variables. And then they need to be documented. So whatever you would change in the data set, it needs to be recorded how you made that change.
Thank you, Joe. We have a question from the audience, I believe.
Again, not so much a question, or maybe a question and a comment. You talked mainly about electronic data, but of course there is still physical data, such as case report forms and maybe questionnaires, etc. And so I know that we're required to store those safely in locked filing cabinets, etc., and access. Do you want to comment a bit about physical data rather than electronic data?
Sure, and that is still very relevant. We do collect information on paper forms. And again, I would just add what you said. They need to be stored in a locked location. There needs to be a plan for the security of physical data. Physical data will often be that which could be more revealing of patients' identification, and we always need to respect and protect the privacy of patients involved in studies. So having a locked cabinet behind a locked door, so there's two locks before you get to the information, and then only the appropriate individuals having keys or access to the cabinet, I think is an important thing to remember. Thank you. There's a question here. Do you discourage using Excel as Stata, S-T-A-T-A, okay, or it also has its shortcomings? So I have to say I'm not familiar with Stata. Does anybody know what it is? So if it's a statistics package where any changes to the data would be made in a programmatic way, I would say it's fine to use. If it's something where you would go in and sort of hand manipulate the data, if you're going to cut and paste in order to reformat the data, I would say it has the same shortcomings as Excel. The real idea is can you trace changes to the data? So if you have your source data, your original data stored, and particularly if you can make them read only, so you're not writing over them, and then you have a program that documents any changes made to those data as you merge them into an analysis data set or manipulate them to create your analysis form. You want to have some traceability on that, and that sort of fully accounts for the history of the data, so you know where it came from, how it changed. Someone asked also to expand on the question of data privacy, and what do I mean by that? Again, I think it means that you can't attribute data back to a particular person. Nobody would have, you do need the data within a person to be uniquely structured, so you have an ID number, but you can't go find somebody's name, you can't go find their address. And so a person's identity is maintained as private once you get to the point of having an analysis data set or a data set that's going to be shared with any other group. You want to, it's really critical from an ethical standpoint to maintain an individual's privacy. So we can use identifiers, identification numbers in order to group their data. We need to be able to keep bioanalytical data together with case report form data and that sort of thing and merge those data sets. So it's not that we don't know what data comes from an individual. We just don't want to have in the data set and in the storage who that individual was. There's another question, the issue on backing up of data off-site. What happens to the multiple data sets all over the place after the completion of the study? So that's an excellent question. A part of your data management plan really should be how are the data going to be stored long term, and where would that be? So you need to have a part of your plan which are backups and which are the original data, and then have a plan for what it will be the long-term storage.
So I would like to thank Joe for taking us through this question and answer session, and I'd like to thank the audience, the live audience here in South Africa, at Strathmore, in Nigeria, and a few of the other places. Just a few closing remarks. This webinar has been recorded, and therefore you will get a recording of the webinar once it's ready, and if you'd like to refer to it or share it with a few of your colleagues who did not attend, please feel free to do so. Additionally, as Pharmacometrics Africa, we would like you to take part in our survey. We are going to send out a brief survey about this webinar, and this really is to improve future webinars. We need to be able to address your expectations better. So please take a few minutes to fill that out when you get the survey. And from us, really, thank you for tuning in. And lastly, please feel free to visit our website at www.pmxafrica.org and get in touch and just follow our activities. So thank you very much and have a nice day.